Okay, so we have here the CD markers or the surface markers. Okay, the surface markers, actually, these are the markers present on the surface of our T and the B lymphocytes that will help us differentiate the different stages of our T and the B lymphocytes. Because under the microscope, it's just look under the microscope, our lymphocytes, we could not distinguish actually as your T and the B lymphocytes. So if you happen to identify the presence of the lymphocytes under the microscope, we tend to report that one as only as lymphocyte. We are not distinguishing that one as either B lymphocyte or T lymphocyte. So the best way to differentiate them is by their CD markers or the surface markers. CD markers are a cluster of differentiation markers. Okay, so this one is being identified primarily by your monoclonal antibody. Because of course, monoclonal antibody try to react specifically to each of your antigen that will help identify the different CD markers as your antigens. Okay, so again, we have here the stages of the development of our B lymphocytes. So we're going to discuss the detailed development, differentiation of our lymphocytes, starting here with your development of your B lymphocyte. So again, as a summary, okay, so the differentiation of your T and the B lymphocytes here will also try to follow the antigen-independent maturation occurring on your primary lymphoid organ, followed here by your antigen-dependent maturation occurring on the secondary lymphoid organ. So for the development differentiation of your B lymphocyte, that one is geared towards the production of our antibody. So gagawa siya ng antibody. As it try to mature, it try to produce our antibody. So again, our antibody, okay, this is a typical Y-shaped antibody morphology. So we have here your heavy chain. So itong heavy chain natin. Because your antibody is made up of your heavy chain and we have also your light chain. Okay, the heavy chain is encoded here in chromosome number 14 for you to produce that one. So, the heavy chain of your antibody could be either mu, alpha, delta, gamma, or epsilon. Each of, the heavy chain here, the type of the heavy chain determine the type of your antibody. Like if the heavy chain is mu, that's your IgM. If the heavy chain is alpha, that's your IgA. If the heavy chain is delta, that's your IgG. This one is gamma, so that's your IgD. For the delta is IgD, for the gamma is IgG. And for the epsilon, if the heavy chain is epsilon, then that's your IgE. On the other hand, another structure of our antibody, we have here our light chain. Our light chain could be a kappa or a lambda. So, either lang sa kanila, it never both of that. Because magiging abnormal na yan pag dalawa. So, the kappa here is encoded in chromosome number 2 and their lambda is being coded in chromosome number 22. So, when speak about the development of your B lymphocyte, so gagawa siya nito ng antibody. Una muna, gagawa siya ng heavy chain followed here by the light chain. Okay, so we'll be discussing the development of your B lymphocytes. So, from your hematopoietic stem cell or your pluripotential stem cell, that would eventually be acted upon by growth factors like your E2A, ABF, your PAX, and we have also here the cytokines. We have in the form of your interleukin 7, cytokine in the form of your interleukin 7. If that one, all of this growth factor here try to act on your hematopoietic stem cells, that would allow the differentiation of your cell here to become your B lymphocyte, starting with the pro-B lymphocyte. So this is the earliest stage in your B lymphocyte development. Okay, the pro-B lymphocytes here, or pro-B cell, contains the following CD markers. So they are found in the surface. So, if this was your B lymphocytes, so pro-B, it contains lahat ito. Okay, we have your c-kit. The c-kit here is the surface receptor. Primarily, we try to interact with the stromal cell or your hematopoietic stem cells that would allow your c-kit ligand to act on that and it would allow it to differentiate. Aside from your c-kit, this also contains your CD19 marker, serving here as a co-receptor for the further development of your B lymphocyte. CD24, CD43, and CD45, serving here as a signaling receptor for further development. Aside from this one, also contains the following, also contains the enzyme TDT, your terminal deoxynucleotide transferase. Another one also contains here the gene RAG, recombination activation gene, which are needed for that to undergo the development. So your pro B cells here try to undergo development by undergoing gene rearrangement in order for that to produce here your heavy chain. The first na antibody na produce niya is the IgM. So this one, gene rearrangement primarily for the production of your heavy chain mu. Okay, so gagawa dito siya ng heavy chain mu on the cytoplasm na sa loob. Once successful siya sa kanyang gene rearrangement for the production of the cytoplasmic mu, it become the next stage, your pre-B cell. Okay, so after the pro-B cells has become successful with the gene rearrangement, in order for that to produce here your cytoplasmic mu, it become your pre-B cell. Okay, the pre-B cells, on the other hand, contain the following markers, CD19, 24, and 45R. The previous markers like your c-kit, your TDT, your CD43 would try to disappear on this stage. On this stage, pre-B cells here try to undergo another gene rearrangement, now primarily for the production of your light chain. Okay, so once it becomes successful here, able to produce your pre-B cell receptor. Okay, the pre-B cell receptor is made up of your cytoplasmic mu, heavy chain, plus your surrogate light chain. Okay, so para hindi pa siya ganun ka-fully na magiging IgM. Okay, so in this stage, is to try to produce pa lang ang ating IgM, but still on this stage, parang immature pa din ang ating IgM na antibody. So, ang may produce na pa lang ay cytoplasmic mu, so may heavy chain na siya. Ang light chain, surrogate light chain, it just begin to produce that one and try to produce here our pre-B cell receptor. Once you try to produce your pre-B cell receptor, okay, that will be expressed already on the surface that serve here as a signal for clonal expansion. So it will serve here as a signal for the neighboring ng mga pre-B cells na gagawa sila ng same type of the antibody sa kanyang ginagawa. Then after that one, so if it has already the IgM, kanina nasa loob lang ang IgM natin, and now pag immature B cell na siya, it eventually try to express your IgM on the surface. Nasa labas na, nasa surface. Kanina nasa loob ito, pag dito pa lang sa pre-B cell na stage, nasa loob pa lang ang IgM natin. Okay, and try to mature, and eventually it will be expressed already here on the surface. So that become your immature B cell. So the immature B cell contains the following markers, CD19, 24, 45R, plus your IgM expressed on the surface. Aside from that one, also try to express other markers. It contains here CD21. Okay, CD21 marker here is needed for the activation of our complement system. CD40 and your MSC class 2 for the interaction with your B and your T cell. Okay, and after that one, it try to undergo your negative selection process. So negative selection process is screening test. Okay, that will check if your B cell here is a functional B cell. The basis for your negative selection process is on the recognition between self and the non-self antigen. So if that B cell could not recognize between the self and the non-self, it will be removed by the process called here apoptosis. Okay, so again, pag self-antigen, dapat ang B cell natin will not react on that. Okay, pero pag non-self, so it will react on that because of that one is your pathogen. So like for example, why are you removing those B cells here were intended to self-react? To prevent primarily our autoimmune disorder. Kaya pag nag-self-react siya, okay, eliminate that one, remove that one, so defective siya. Okay, remove that one by the process of your apoptosis. And that's occur here on the immature na stage. Okay, the next stage, we have the mature B cell. Okay, so kanina, from this stage, okay, this one occur in our bone marrow. Okay, then eventually try to mature. Sa bone marrow, nag-mature na siya, and eventually pupunta na sa ating secondary lymphoid organ. Okay, your mature B cell would be differentiated here from our immature B cell because it try to contains not only your IgM on the surface, but also try to express another antibody in the form of your IgD on the surface. Still, it try to retain other surface markers, CD19, 24, 45R. Okay, para saan ang IgD, an antibody here on the surface aside from your IgM? So the IgD here helps primarily with the interaction with your antigen. In the presence of your IgD, it will try to prolong here the lifespan of our B cell. Actually, the IgD here will try also to decrease the ability of your B cell to combine or to react with your antigen. Okay, so ito magiging consequence if your B cell here will able to meet with your antigen. Okay, it would have eventually a longer lifespan, but if that one cannot able to meet with your antigen, it will die easily. Okay, ulit. Okay, immature natin ay nasa bone marrow ito. And then try to mature. After undergoing here the negative selection process, try to mature. And then try to migrate to your secondary lymphoid organ because since mature na siya, it's already ready to meet with your antigen. Okay, so ito na siya. If you're mature B cell, so it would have here the IgM and IgD on the surface. Okay, so after you have your mature B lymphocyte here, so the next stage we have your activated B lymphocyte. Activated B lymphocyte here would just differ with our mature B lymphocyte by the addition of another marker, which is your CD25. So these are your mature, di ba, na B lymphocytes made up of your, aside from your CD19, CD24, CD45R, aside from that one, also try to express your IgM on the surface plus your IgD. In addition to that, our activated B cell here would eventually express also your CD25 marker. The CD25 markers serve here as the receptor, which allows the interleukin-2 coming from your T cell. So this is being produced by your T cell to bind in your B cell that basically help your B cell to develop here and eventually helping it to produce your antibody. Okay, so upon meeting up with your pathogen, so what we're going to happen here with your activated na B cell? It could either transform to become your plasma cell or it could also be your memory cell. Okay, your plasma cell measures approximately 10 to 20 micrometer in diameter. This one would have an eccentric nucleus with the presence of your perinuclear halo or hoof. And then, this is also characterized by an abundant cytoplasmic immunoglobulin. So, madami siyang immunoglobulin on the cytoplasm. There are few immunoglobulin on the surface. Okay, this is your plasma cell. This is your antibody-forming cell. So, this is being destined here primarily to produce your antibody. So, once it tries to produce antibody, it dies. So, therefore, we consider here plasma cell as your antibody-forming cell. Or another option for your activated B cell here become your memory cell. So morphologically, your memory cell resemble your inactive na B cell, only that would have here a longer lifespan compared with our inactive B cell. And the memory cell here is being programmed to remember, okay, the antigen previously para pag next encounter, madali na siyang i-remove ng ating B cell because this one has already been recognized because of the production of your memory B cell. Okay, now we go to your development of your T lymphocytes. So again, the development of our T lymphocytes also follow here both our antigen independent and antigen dependent maturation process. Okay, so we have here the stages of the development of our T lymphocytes. So from your hematopoietic stem cells that was being acted upon by your growth factor, specifically we're talking about the cytokine interleukin-7 that will convert your hematopoietic stem cell to your thymocyte. Our thymocyte here is a double negative. So CD4 negative and CD8 negative. So hindi pa siya mature. Once you have already your double negative, it try to undergo gene rearrangement by combining with your CD3 plus the beta globin chain. So once your beta globin here try to combine with your CD3 positive, it become your pre-T cell receptor. Okay, once you produce already here your pre-TCR receptor or T cell receptor, this pre-T cell receptor try to signal here your thymocyte is double negative to undergo transformation to become your double positive. Okay, this is your double positive na. And then this one try to undergo another gene rearrangement for the production of your full mature na T cell receptor. So para makapag-produce tayo ng mature na T cell receptor or complete na T cell receptor, so your CD3 plus beta globin chain, you need to combine your alpha globin chain. So once your CD3 plus your beta globin chain plus your alpha globin chain combine, that become your T cell receptor, which is already a mature and complete T cell receptor. So receptor ito ni T cell natin. Okay, so we have here the double positive T cell here. So when speak about the double positive, that's already considered to be the mature one because that already can be differentiated already into your CD4 positive. Other name of your CD4 positive, you have your T helper or your T inducer, or the CD8 positive, that's your T cytotoxic or your T suppressor. We have here the relative concentration. So again, T helper is approximated as 2/3 of the entire population, and then your CD8 is 1/3. So 2 is to 1 ang ating ratio. Mas madami dapat ang helper compared with your cytotoxic or your suppressor. Okay, then after that one, once you have already double positive here with a full TCR receptor in the surface, this one try to undergo a selection process or a screening test. Okay, so first it undergo here the positive selection process. The positive selection process here would try to select your double positive pair in its ability to differentiate or to identify their respective na major histocompatibility complex molecule. So if this one's your CD4 positive, it should identify your MSC class 2. If this one's your CD8 positive, it should recognize your MSC class 1. Pag hindi niya kaya mag-recognize, tatanggalin siya sa ating immune system natin, and try to die by your apoptosis. Followed here by your negative selection process on the basis of distinguishing here between the self and the non-self. Again, pag self, dapat hindi siya mag-react, and the non-self, dapat mag-react siya doon. And though, pag hindi niya makilala between self and non-self, then also try to be removed by our apoptosis. Only that, ito na lang matitira. 1 to 3% of your double positive here which are functional. Functional respect that it will have here the ability to recognize between your MSC molecule here and also differentiating between yourself and a non-self. Okay, then we have here your maturity cell. So this is your maturity cell already. So your CD4 positive. Again, your CD4 positive, that's your T helper. Other name for that's your T inducer. So our CD4 positive T helper can recognize our antigen or your pathogen if that one is being associated with your MSC class 2. Ito si antigen, pagkasama siya ni MSC class 2, makikilala siya ni CD4 positive natin. This is your T helper, ang kanyang receptor dito is your CD4 positive. The CD4 positive can recognize the antigen if your antigen is associated with your MSC class 2. And therefore, it can kill this one, can remove. Pag hindi ito, if you have your antigen, tapos wala siyang MSC class 2, it will not be recognized by our CD4 positives or T helper. And therefore, could not remove that one. That's why kailangan nila ng MSC or the major histocompatibility complex molecule for them to be recognized. Now we go to the subsets population of your T helpers. So we have your T helper 1, primarily produce the following substances. We have your gamma interferon, we have your tumor necrosis factor beta, which are involved in the intracellular removal of our pathogen, provided that one again is in association with your MSA class 2. T helper 2, try to list here the following cytokines, interleukin 4, 5, 10, and 13, which helps here in the antibody production of your B cell. And T regulatory, try to regulate the balance between your T helper 1 and T helper 2 to prevent here your autoimmunity. And another one, we have your CD8 positive. This is your T cytotoxic T suppressor. So this one, try to recognize the antigen of this one has been associated with your MSC class 1. Basically, your CD8 positive here or the source T cytotoxic suppressor can kill specifically your virally infected cells. So, cell na na-infect ng virus or your cell is a tumor cell provided that one is in association with MSC class 1 para siya mapatay. Pag hindi, pag wala ka MSC class 1, it will not be removed or killed by our T cytotoxic. Okay, next we go to your activated T cell here. So activated T cell expressor CD25 and also try to serve here as a receptor for the interleukin 2. So once your T cell here eventually try to meet with your antigen, it becomes sensitized. Okay, so on the next encounter with the antigen responsible for its sensitization, it try to release here your cytokines. And once it try to release the cytokines, okay, that would eventually be it will be manifested here as your cell-mediated immunity. Once you try to release the cytokines released by your T cells here will be involved in our cell-mediated immunity. Again, magmi-meet muna siya ng antigen. During the first encounter with the antigen, it try to become sensitized. On the second encounter, it try to release the cytokines. And the cytokines it try to release will be involved in our cell-mediated immunity. Again, what are those cell-mediated immunity natin or reaction? Tumor killing, again, it can kill directly our tumor-infected cell or virally infected cell. In the case of your T cytotoxic, provided that one has been associated or your tumor or your viral infected cells being associated with your MSC class 1. Okay, your T cytotoxic here is also involved in your graft rejection or rejection of your transplantation. Another one also, the cytokines released here by our T cell, especially your interleukin 2, okay, helps here in your antibody production. It can help in the development of our B cell for the antibody production. Okay, the interleukin also being produced by your T cells, so interleukin 2, okay, try to help also with our hematopoiesis. If I try to check our hematopoiesis, so may mga growth factor tayo doon, interleukins like interleukin 2, which help para magkaroon ng differentiation ang ating hematopoiesis, and that one is being produced by our T cell natin. Another one, so the T cell also involved here in our delayed type hypersensitivity reaction is a type of your cell-mediated immunity. Okay, now we go to the next. We have here the third population, lymphocyte. We have here your NK or natural killer. So natural killer here try to possess receptors which are not found in your T and the B lymphocytes. This is your third population lymphocyte. This is your large granular lymphocytes. Usually, di ba, ang ating lymphocytes they are agranular, but this one is granular. Okay, so for the development of your NK that try to occur here in our primary lymphoid organ, the bone marrow, and that one is under the influence of your interleukin 15. Di ba, your B lymphocytes also try to develop here primarily in the primary lymphoid organ, which is your bone marrow, but that one is under the influence of your interleukin 7. But this one is interleukin 15 for the NK. Okay, for the surface receptor of our NK, so there are two identified here. We have your CD16. The CD16 receptor of your NK is the one we try to bind on the FC portion of your IgG antibody. So this is your antibody, try to coat your pathogen. So if your pathogen has been coated here by the antibody, so that's your opsonization, your NK can bind on this antibody-coated na pathogen, and your NK can kill this one. Okay, you call it once your ADCC, antibody-dependent cell-mediated cytotoxicity. It can kill your tumor-infected cell. It can also kill your virally infected cell, provided, however, here that your tumor-infected cell or your viral infected cell is being coated by your opsonin, the form of your IgG antibody. Because again, ang NK natin, hindi siya makarecognize ng pathogen to the CD16 pag wala kang IgG antibody because the CD16 na niya na surface marker would have receptor dito sa FC portion ng ating IgG antibody. So ano na-notice natin? Pareho sila ng activity ng ating T cytotoxic. Your T cytotoxic can kill your viral infected cell or tumor infected cell, provided here that your T cytotoxic requires MSA class 1. Ito naman, kailangan mo ng antibody. May nakakode dapat sa kanya na antibody sa ating viral infected cell or tumor infected cell in order for your NK to kill that one. Another marker, we have your CD56, but as of today, hindi identified. Unknown ang function nitong CD56 marker in your NK. Okay, so we have here the subset of the population of the NK. So you can have here the NK population wherein it would have a high level of the CD16 markers and low level of CD56. So CD16 marker will always be the surface marker for the able to bind with your IgG antibody that would exert its cytotoxic activity. So it would have more cytotoxic activity. On the other hand, the population would have a higher CD56 marker compared with the CD16. Those are the NK wherein it try to produce here primarily more cytokines, like for example your gamma interferon and your tumor necrosis factor beta. Tumor necrosis factor alpha. Okay, then we have here the mechanism of cytotoxicity of your NK. So how your NK were able to recognize what cell are going to destroy or not to be destroyed. So it would be highly dependent on the presence of your receptors. In the presence of your inhibitor receptor, so pag sinabing inhibitory receptor, that one is being possessed by our normal cell or by self-antigen. And therefore, pag inhibitor receptor ang ma-recognize ni NK natin, it will not kill or destroy that one. So, example for that, we have your MSC class 1, your KIR, killer immunoglobulin receptor, ILT, LIR receptor. On the other hand, if your NK try to recognize that the receptor possessed by the cell here is activatory receptor, mean to say that one is not yourself, mean to say that one is a pathogen. So, it will be destroyed by our NK. So, pag hindi sila nag-possess nito ng mga inhibitor receptor, so pag wala ito, hindi ito makita ni NK natin, so that cell here will be destroyed by your NK. So, those are tinatawag natin na mga activatory receptors. So, example for that, mga ito. Plus, pag wala siyang nakita ng mga inhibitory receptor. Okay, so primarily the cytotoxic activity of our NK because of number one, the presence of the perforins. The perforin here is a channel protein that try to produce here the hole on the surface membrane of your cell. And we have also here the granzyme. So this is the enzyme responsible here for the cytotoxic activity, responsible here for the lysis of your cell with respect to the cytotoxic activity of your NK. Okay, and lastly, deficiency of your NK, if the patient would have a low level of the NK, so the patient would have a recurrent viral infection, pabalik-balik na viral infection. So that's how important your NK inside our body as part of your immune system. Okay, so we have here the difference between your T cell and your B cell. Okay, so the primary lymphoid organ of your T cell is your thymus, and for the B cell, we have here the bone marrow. The T cell can populate in the peripheral circulation, thoracic duct, or even your lymph nodes, whereas your B cell can be found only on the bone marrow, your spleen, and your lymph nodes. And then your T cell here is capable of the formation of your rosette pa siyang rose, di ba? With respect to your shaped red blood cell. And of course, your B cell, this is your antibodies try to express immunoglobulin on the surface. Another differentiation would have here the CD markers. So CD2, 3, 4, and 8 are found on your T cell. And for the B cell, that's your CD19, 20, 21, 40, and we have your MC class 2. Okay, so thank you.